if you could text him the number. What I might do is, if it is 58, I mean, actually, stop the Facebook and I'll start the Facebook again, because then we can delete that other stream. Stop live stream. Okay, that's a good placeholder. Hey, Lillian, how are you?
Hey. Hi, Lillian.
Thanks for joining.
No worries. I was just in the 15 minutes before the presentation session and nobody joined in there.
I think we were just trying to figure out whether it's, were you trying to join the other one or here?
The other one.
Yeah, sorry, I didn't share that. Yeah, we had set up as a placeholder there. But... Hey, Fabien, thanks for joining. Hey, Neeraj, thanks for joining as well.
Hi, everyone.
Hi, Neeraj. Hi, Fabien.
Hi.
Hi.
We have pretty much everybody here. I think we, Alex, whenever you're comfortable, all yours.
Sure. So this is the FOXG1 Research Webinar, discussing current trends and the importance of research. Hi, everyone. Thank you for attending our first webinar. This afternoon, we will be discussing disorders, the research of disorders, what new technology is available to study these trends in research, and why it is important for research to continue in these areas. We will have two guest speakers from different perspectives in the research world. Firstly, Dr. Fabien Delerue, whose research focuses on genetics. A misconception is that genetic disorders are not that common and only affect a small part of our population, such as those with trisomy 21 or more commonly known as Down syndrome. However, many diseases have a genetic component, such as cancer and Alzheimer's disease, which affect a large percentage of our population. In this session, we will hear from Fabien all about how he is using animals to investigate the mechanisms of FOXG1 syndrome and how changes in our genes can explain and relieve other genetically based disorders, such as dementia. On the other side of the spectrum, from our second speaker, Neeraj Kothari, he looks at medical data, which is produced from people. His company analyzes this data and uses artificial intelligence to expand our knowledge of human disorders. He will explain how important this data is and how bioinformatics is the future. Finally, we will hear from Vivek. He will give us some perspective into what it is like to be a father of a child with a rare genetic disorder. Vivek is the founder of this foundation and heartwarmingly started this foundation after receiving the diagnosis of his own son to have FOXG1 syndrome. The aim of the FOXG1 Foundation is to raise money to fund research into finding a cure for FOXG1 syndrome and how to understand such a tiny error in the genetic code can have such dramatic consequences to one's health. This is our agenda for this afternoon. The whole session is projected to run for under 45 minutes. General housekeeping rules are that you keep your mic on mute when you're not speaking. Please hold on to your questions or type them in the chat throughout the presentations. We'll let you know when it's a good time to ask your questions. Additionally, we will welcome you to have your cameras on during the session, as it will give the speakers a sense of speaking to a real audience, something that we have been deprived of in these COVID times. So, hi, Fabien and Neeraj, welcome. Could you please give us a brief introduction to yourselves for about a minute each? When you're ready, Fabien.
Sure, thank you very much, Alex. So my name is Fabien. I've been working in neuroscience, particularly Alzheimer's, Parkinson's, so dementia as a whole for the last 25 plus years. But over the last 10 years, what I specialized in was genetics, and it has happened pretty much as I went. The rationale for it is when you have disorders like FOXG1 or others that have a genetic component, the first thing you need to do, and I think Neeraj is going to talk about that even more than me, is actually identifying what the genetic cause is. And once you know what it is, I think Vivek will be well placed to speak about that as well, because the journey as to the identification of the FOXG1 mutation was certainly quite a long one. But after that, the idea is also to be able to reproduce such mutations in animals or in cell models, and obviously to try to apply a gene therapy where you reverse the mutation. And for that, you need really high-end tools such as next generation sequencing. So that's what I've been doing in a nutshell for the last 25 plus years.
Thank you, Fabien. Now, when you're ready, Neeraj.
Hi, everyone. Really excited to be here. And thanks, Vivek and the group for putting together this exciting little foundation. And hopefully with time, it will grow into something much more bigger. So I'm co-founder and chief technology officer of Cogniant. And I've had a background in software engineering, data science, and data engineering in last 12 years. Before starting Cogniant, I was part of Singapore government's Ministry of Health task force looking at creating their national analytics strategy around bioinformatics and healthcare technologies, and using digital health as tools to monitor large cohorts of population. My interest through Cogniant has been, and this was genesis of Cogniant, is to look at real world evidence of patients of neuroscience. So there is a large scale research, and currently it's very growing field, like Fabien mentioned, of genetics. And since human genome had been mapped, there is increasing activity. So what we want to combine is phenotypic data to this genotypic data, looking at what are the real world phenotypic implications of different disease modifying therapies, or even disease itself to start with. So we are highly focused on bringing digital approaches to measure this phenotypic data before, during the onset of the disease till the treatment and monitoring of the disease when they're undergoing the therapy and treatment. Yeah, so that's me.
Thank you, Neeraj. So yeah, thank you both for those introductions. Now let me hand over to Poonam, who is going to explain the macro landscape, like what it is, what's happening in it, and yeah, go ahead, Poonam.
Thank you, Alex. Thank you, Fabien and Neeraj, and welcome everybody who is attending the session today. As Fabien and Neeraj explained, like what their expertise is, and so with their expertise, we can just get a glimpse of the current trends in research, and because they're already working on some of the latest technologies surrounding health and medical research. So just to give you a macro view in this space, I'll be talking for a few minutes. So I think it's pretty imperative to look at how technology is impacting the way therapies are being developed at a much faster rate, at a much accelerated rate than before. Though the pharmaceutical industry has been relying on cutting edge technologies to help deliver safe, reliable drugs to the market for a very long time now. So the use of technologies such as artificial intelligence and machine learning has it has a lot of intelligent applications such as diagnostics, digital therapeutics, digital biomarkers, such as Neeraj is working on, helping identify patients for clinical trials, accelerating the drug discovery, manufacturing, and also in predictive forecasting. So these, there are a lot of applications, and this has been disrupting the entire process and how things were done traditionally. So to start, to speak about the startup ecosystem in Australia, so there are a lot of companies which are really working in the life sciences space, not just in Australia, but also worldwide, as you might have heard of DeepMind, and there are a lot of other companies who are working in this space. A lot of digital health startups which have come up, and they are doing a lot of disruptive work in this space. And also there are a lot of experts who are continuously talking in the digital health space in social media, such as the medical futurist. Yeah, so there are a lot of things happening in this space, and I feel that with these kind of technological advancements and a need to find cure for some of the disorders for which there's no current cure available, I think we can, for sure, we can work together and connect the dots, working in collaboration with different organizations and researchers, and work towards finding a cure. And this space definitely looks very bright, and the future looks really bright in this space. So thank you. And
yeah, thank you so much for that insight, Poonam. It's something that it's really exciting, but I'm yet to learn about it. Now it's time for Fabien's presentation. Fabien already introduced himself just before, but Fabien's group, or Fabien and his PhD student, were the first research group to receive a grant from the FOXG1 Foundation. He's going to explain the kind of work him and his previous student, Daniel Tan, did towards FOXG1 research, and the story of how this came about. As much as you would like, Vivek can fill in the gaps. Go ahead, Fabien.
Thanks, Alex. Yes, so I forgot to mention I'm currently at Macquarie University here, but since I moved to Australia, I work for 3 different universities. I started at Sydney Uni, then we moved at UNSW when the FOXG1 project started, and I got to meet Vivek. And then in 2018, we moved to Macquarie University, where the project actually ended up quite recently. So because we are about to publish the research that we carried out for the last 3.5, 4 years, I won't be able to share the slides, but I can definitely bring you towards the journey, because it has been a journey as well in terms of research. So as Alex mentioned, the first thing we did when we met with Vivek was try to understand what was the research landscape. I'm talking about 4 or 5 years ago. And quite frankly, at that time, there wasn't any research going on. There was barely anything. And that is due to the fact that FOXG1 syndrome is a word and a diagnosis that we now put quite easily on children and families, with all the burden that it implies. But let's not forget that only 5 to 6 years ago, even the word wasn't, FOXG1 syndrome was actually almost not existing. And that is because, thanks to the technology that, for instance, Neeraj talked about a few minutes ago, then we have been able to actually identify FOXG1 syndrome. 10 years ago, kids who were suffering from FOXG1 syndrome did not have the luxury to even have a diagnosis. And that was has been developed with what I touched base a minute ago, which is next generation sequencing, for instance, and I will leave that to Neeraj. But let's put it where it was. So let's say 5 years ago, kids like Kush had a diagnostic, and that was FOXG1 syndrome, which implies that they carry a mutation in one of their genes, which is called FOXG1, and that induces this debilitating disorder. Now, the thing is, when no research is carried out anywhere, the first thing you need to do is to develop animal and cell models. Why you need to do that? Because these are tools that are essential to give teams like mine the ability to explore actually what's going on, because the disorder is so rare that no one actually knows exactly what's happening. So the first thing we did, because as I said, I specialize in genetic engineering, was to create a mouse model, the first mouse model of FOXG1. There were a couple of mice that were related to FOXG1, but not what I call a clinically relevant model. What does that mean? That means that we had the ability to look at the mutation that Kush is actually carrying and reproduce the exact same mutation either in cell lines or in the mice. And as you can imagine, that is a brilliant tool afterwards for researchers to actually study not only what's happening at every level, genetic level, cellular level, but also biochemical, or as well as physiological, so the whole animal tissue in cell culture models, for instance. And with these tools, then you kind of build an entire story and you start digging in and trying to understand. So the reason why we've been able to do that is because, as Alex mentioned, the Australian FOXG1 Foundation funded my PhD student, Daniel, who is now back to Singapore, but for 3.5 years, he's been able to characterize the cell and the animal models, so the mice and the cell lines. And he did a battery of tests. I won't mention all of them, obviously, but to give you an idea, one of the first one that we did was, for instance, using the newly generated mice and do what we call RNA sequencing. So in short, RNA-seq. This technology that Neeraj is probably very familiar with allows you to take a mouse that does not carry any mutation and look at how the genes are expressed in these mouse. And then you do exactly the same with the FOXG1 mouse, and you see the difference between those two mice. And that informs you and tells you well, what can possibly go wrong? So these are really valuable information. This was done at the RNA level, but you can do exactly the same at the protein level. So we carried out what we call Western blots as well on these mice and on these cell lines, on these FOXG1 cell lines, and eventually, when you put all the technology together, so at the DNA level, that is your ability to sequence, so to read the entire genome, at the RNA level, I just mentioned RNA sequencing, or at the protein level, which is the ultimate level within a given cell, for instance, your neurons, your brains. And also the ability, we now had the ability to observe the mice. Neeraj was mentioning phenotyping. It's a scientific word for saying you observe what the behavior of the whole animal, of at the whole organism level, what this behavior is. And once again, you can compare a mouse that does not carry the mutation with one that does carry that. And we did a battery of tests. When I say we, I mean Daniel did that. And we observed some differences in which were quite significant. In a sense, they were recapitulating the same indications that you would find in FOXG1 children. So we were really happy about that, because although it was not easy to identify in the first place, simply because the mouse is not a human being and vice versa, we still had some common grounds that allowed us to eventually put all these data together. And then we came up with something really promising, because we have one candidate, one gene, one protein, that we think is really important in what's going on when the disease kicks in. So we hopefully should be able to publish that before the end of the year. But obviously, this is a really small part of what needs to be done when we're talking about finding a cure and bringing it to the children who desperately needs it. And the reason why I'm saying this is, although I'm really happy with what Daniel did, it is not always the case that you have a PhD student that's able to bring an entire story from the beginning to the end and have a candidate, which I considered a very, I'm talking about the candidate gene, that would be a target afterwards for the clinic to test this candidate. But that's when we also need the support of other people. For instance, we need the support of clinicians, of industries, who will be able to take on board the information that we bring through publications, and then bring this information to the next level, which is indeed carry out clinical trials and trying to develop both gene therapy, which is a way of correcting the mutations in the patients, but also more traditional approach that are based on pharmacological, like I'm talking about drug delivery. And all this needs to be tested. The reason why I'm saying that is because it is not sufficient. It is not enough to have, for instance, a candidate which actually may or may not work when you target it. You also need to have tools such as how do you actually deliver the drug or the gene therapy to target your gene or your protein of interest. Let's not forget that FOXG1 is essentially expressed in the brain. So it is not as easy as delivering a drug to the bloodstream, for instance. So we need tools to deliver these new therapies to the brain of the children. And that requires testing, that requires clinical trials. This is why we still need to continue the research at every level. We need funding for that. And that's where we are up to in my small team. but I think and it seems to be the case, all the families come together, the community come together, but also different partners, being governmental partners, and also private companies, then I think we have a chance to make a difference and indeed eventually find a cure for these children. And I think I will finish on a note that that is even more optimistic. When we do a small project like that, which gets me really excited, but probably seems very small compared to the scope of what is needed for these children, the knowledge that we gain when we do that is also benefiting other rare genetic disorders. And I think this is what's really important at every level. The families come together, but also the organizations come together. FOXG1 is associated with other rare genetic disorders. And there is, particularly in Australia, an organization that's called Rare Voice Australia. And I think the strategy behind it is really promising. If everyone comes together, then we'll be much more powerful and in a better position to fight conditions such as FOXG1 syndrome. So I don't know, maybe I've been a bit over time, I'm sorry if I did, but that's about, yeah, the journey, the research landscape and the really what we've achieved and what still needs to be done. But I think we now go back to with Neeraj's presentation to another very important part of the journey. As I was mentioning, rare genetic disorder as a whole, I believe what Neeraj is doing is also helping identifying new rare genetic disorder. We have many children and patients around the world who still don't have a diagnostic. Actually, most of the rare genetic disorder do not have a diagnostic. And that's where people like Neeraj can really make a difference. So thank you very much for listening. Thanks again for the invitation. And I'd be happy to take questions at the end, obviously.
Yes, thank you, Fabien. I was going to allow for an intermission here, but I might not. Yeah, that's absolutely fascinating. I think that kind of research and that
Alexandra, I'm happy to take any question. If I don't
If anyone wants to ask questions, you can now, or you can write it in the chat. But I think that's fascinating because it's going to change how we approach therapies in the future completely, like pharmacologically and everything. Yeah. So. Oh, sorry, Vivek. I didn't see your thing because I'm screen sharing. Does anyone have any questions for Fabien? I'll allow a minute for this.
Not a question, just a comment. Really impressed by the work your group did, Fabien. So really interested to see the publication when it comes out. So yeah.
Thank you, Neeraj. Yeah, this was quite a successful PhD project. As I mentioned, it's not always that impactful, but I think both Daniel and I considered that project really as something very different than what we used to do, because when you used to be working on, what I call mainstream pathology, which are Alzheimer's, Parkinson's, this heavily funded kind of disorder, when you're approached by a family, in that case, Vivek and the kids, and you really can have an idea of you have a tangible idea of why you're doing what you're doing. I think that's also why this project was actually successful as it as it has been. So yeah, we were quite happy with that for sure. And yes, I can't wait to publish it as well, for sure.
Yeah, absolutely. It's very powerful.
Fabien, if I may, when we were celebrating the conclusion of the research project, Lars did mention that it was in the top 1% of the projects. Like, it was very impressed. I think the lab heads were quite impressed by the outputs. Would you be able to maybe give a slight, very brief layman's explanation as to why this was so different? And maybe as we're discussing the current trends, so specifically monogenic, they have a bit of a slightly higher probability than disorders that span across multiple genes. Did you want to touch base very briefly on that?
Yes, so it has to do with what you mentioned, the fact that somehow we can pinpoint. We started with the starting point was we knew the culprit. We could target FOXG1, which is not, and as I mentioned before, it is not always the case. Many of the disorders do not have even a genetic cause that has been even identified yet. But having said that, it was a blank canvas, really. We had absolutely no idea. So I think that the reason why the project, in my opinion, was so successful, I'm talking about the PhD, Daniel's PhD project, is twofold. First, because it's full credit on Daniel, because Daniel was a really experienced PhD student who was hardworking and really driven by the project. And the second thing is that Daniel used and mastered a lot of different technology, and it is not that easy. Usually you got people who specialize in one or two different technologies and master them quite nicely, and they really become expert at this. But rarely do you have a person who is able to understand, master, and produce results out of many different tool sets and techniques. And I think that's why that's what was implied to be able to build an entire story. So I think that's why ultimately the project was in the top 10% for sure.
Thanks, Vivian.
That's great. Congrats to Daniel. I hope he's doing amazing things in Singapore. Okay. Yeah, thanks everyone for contributing. Now on to Neeraj's presentation. Poonam previously gave a brief explanation of the macro landscape. Now Neeraj is going to go into the details. Things we would like to hear about from you, Neeraj, are about dementia research you've been doing, and additionally the importance of cohort data and what future wins are being worked on. When you are ready, Neeraj.
Yeah, thanks. So for this presentation, I've put down a few slides, if I may share my screen. What I'm gonna share is the work we have done, starting with depressive disorders. So at Cogniant, we are focusing on few different disorders of brain-related disorders in neuroscience. And I'll initially set the context for digital phenotyping or digital biomarkers, and how we are being able to apply that in depression, and how the future might look like for certain rare disorders like FOXG1 syndrome. So we are clinically validated patient-led digital biomarker company. And if you look at current state of the art in diagnosis of many neuroscience condition is largely battery test, question and answer. So this is sample form to diagnose depression. And this is current state of the art diagnosis, which is answering 9 questions based on which a clinician determines you if you are depressed or not. It's really archaic, is not, the neuroscience research hasn't produced a clinically sensitive or highly accurate diagnostics test. It is very subjective to the patient. It's oftentimes lab or clinician administered, and always, often or not, there is a subjective bias in this test. And Fabien mentioned about genomics entering this new era of precision medicine, but a lot of these neuroscience or brain health related disorders are still stuck in this old paradigm of assessments based on question and answer. And we at Cogniant thought, well, what could be a better way of looking at objective, more data-driven approach of understanding and assessing these conditions. And we came up with this knowledge, or with this hypothesis that everyone in the world is digitally connected. More people have smartphones now than access to running water or plumbing. More than 3 billion people own a smartphone. And that's a really effective way of reaching really large scale population, but also getting fingerprints of individuals and how they exhibit or manifest with certain disease conditions, and just capturing that to understand not just their symptoms, but their physiological and behavioral symptoms in the real world. So Fabien mentioned about capturing cellular tissue level biochemical data from mice, but what we looked at is the behavioral and the physiological data of the individuals, which was low cost, very scalable, and didn't require us to didn't risk the person we are measuring, didn't pose any health risk. So the risks were relatively low as compared to, let's say, a gene modifying therapy, but it was also highly scalable. So what we built is our digital biomarker measurement platform, just by capturing mobile phone sensors, and wherever necessary, combining that with 1 or 2 wearables to understand physiological, behavioral data from everyday access smartphone, from activity to their location to cognition, their interaction of the tech, their sleep-wake cycle. And this, putting that together in the context of the disease, before they've been diagnosed till they get diagnosed, and then once they go through the treatment, how this is changing, creating the timeline view of that. So you're able to successfully demonstrate results from our depression trial, where for adults, they were prescribed this smartphone app by their clinicians in Singapore. It's the largest psychiatric hospital in Singapore, where the clinicians onboarded these patients on their platform. And it not only provided rich sense of support and engagement to the patient, but also was able to collect data which clinicians could take actions on. And what we found is these 5 features, like location, movement, their clustering of location, and their randomness of their physical behavior, were strongly correlated with their depression score. So just by using smartphone data, we were able to classify people with depressive symptom or non-depressive symptom with 75% accuracy, which was really useful for us to just get the sense that this is really powerful way of objectively visualizing a disease condition, and also tracking it through as the disease progresses, or as the treatment progresses, if it is worsening or if it is getting better, to understand if your therapy is being effective or not. And since then, we've been able to successfully spread this across the full person's profile. So not just looking at sleep, but also looking at mood, cognition, depression, and mobility, and a lot of neuroscience disorders, try to measure physiological symptoms in that, creating a full patient picture, and also creating objective ways of analyzing the behavior. And in some cases, like Fabien mentioned, even in Alzheimer's disease, there is still a large scale research going on diagnosis Alzheimer's early, and what can we do to diagnose these conditions early, and using some of these technology. I think we have a very promising way ahead. What we are doing, we are aggressively validating it in clinical trial setting so that the biomarkers we end up creating are clinically validated, so that it can be applied in the real world. So we are working with different partners. Currently, we have a pipeline of products we are developing for measuring depression, chronic pain, mild cognitive impairment, working with hospitals in Singapore, Monash University in Australia, and also have few collaborations in pipeline. But yeah, that's in short. Just wanted to highlight the possibilities of applying digital biomarkers from early onset or diagnosis till the treatment and providing care. And I'll be happy to take the questions.
Thank you, Neeraj. I might let Vivek talk first, and then when you finish, you can ask questions together. So, yes, thank you so much for that. That's really interesting. And I'm sure, like you said, that how Fabien was happy. He's excited that that field is going to develop. I'm sure your field is going to develop as well, along with, you know, technology. As technology progresses, you know, Apple Watches say that they have blood pressure monitoring now. You know, it's endless, the possibilities. So yeah, Vivek, it's your time to share your experience. Please explain to us from a father's perspective of a FOXG1 child named Kush, and what it's like to have a child with FOXG1. Why a cure for Kush is important, and what the limits of healthcare are as we know it. When you're ready, Vivek.
Certainly, Alex. Thanks, Fabien. Thanks, Neeraj, for sharing your experiences as well. And thanks, Alex. Thanks, Poonam, everybody, for all you're doing. I think I was thinking of like maybe the five points I wanted to share very briefly. You know, just leading on from what Fabien said and from what Neeraj said as well, and Poonam's point. So I think the first one was that, like, you know, if we look at the iPhone, if you look at Facebook, if you look at Tesla, I was listening to a presentation by Mustafa Ronagi, the CTO of Illumina, the gene sequencing company. So, you know, he was saying that, you know, there was a time where before the iPhone came out, nobody knew that how iPhone would change technology. I think similarly, like, you know, Fabien, we've spoken multiple times before CRISPR came out, like, you know, some of these things would have been absolutely impossible. So maybe even if I take one further step back to the human aspect of the FOXG1 syndrome, I think we've been trying to share Kush's journey. It is without a doubt, it's not a small disorder. It's not a low interruption. It's not a condition that kids can live with. So we definitely, because he's so profoundly impacted, I think there is such a great need to intervene and apply intervention here. And when we started looking at things, the first discussion that led up to a conversation with Fabien just gave us a lot of hope that a cure was possible. So I think that disruption that's happening in technology is absolutely phenomenal. So sometimes we forget that, you know, when we say that iPhone or Tesla or Facebook, you know, even Neeraj, what you're talking about, there is a lot of data storage facility, technical infrastructure, data capture activity and all that's going in, which makes this possible. So that's I think an insight I wanted to be thankful for. The second was, you know, Neeraj, you definitely mentioned clinical versus genetic test, or clinical versus databases for some of the these even diagnoses. So I think with the genetic basis being the lowering of the genetic basis of testing, certainly, absolutely, getting a diagnosis is going to become a lot easier for a lot of people. Fabien, we were fortunate enough to go to a Society of Neuroscience conference together. And if you look at even some of the vendor presentations over there, I think not only is the technology enabling a lot of data, but I believe that if you look at the vendor stands, there is industrialization of, I think, the genetic tools, which is phenomenal. Like, we do not understand it, but you can go on the internet now and order a FOXG1 gene, and it'll be sent to you tomorrow via a FedEx parcel for $30. So there's a full industrial complex that's being built behind this genetic enabling space. I think, Neeraj, just one aspect of the wearables, Kush, he just came out of hospital, came out of a 10-day long stay in the hospital, and we were worried about his stats and all. He's got a Fitbit. He's wearing a Fitbit charge that's got a blood oxygen monitor. So his breathing was shallow and all, but I could just flip his screens and see that, oh, the oxygenation is at 98%. So we don't have to worry too much. Otherwise, if we didn't know how profoundly or when the intervention was needed, I think some very accurate data coming from even consumer grade wearables is so good. And the last thing I wanted to share was that the fifth item was that Fabien, when my first introduction to research, we did say that why can't this be done so simply? But then the quality of research, like a PC2 lab with the apparatus that is what do you call it? In full calibration, everything is the reproducibility of research, the discipline around the infrastructure. If you just walking into a lab for someone like myself, who as a parent, as a non-scientist, it just is so reassuring that there's so much good controls around getting good outcomes. So I think a combination of a lot of these factors, they do give us a lot of hope that while what we're trying to, the problem that we're trying to solve is very difficult, but given that there's so many advances happening, I think, and even if there's half a chance, we should really try our best. So super grateful.
Yeah, thank you so much, Vivek. Thank you for everyone for participating in this webinar. Special thanks to Fabien, Neeraj, and Vivek as our speakers, and Poonam as my co-host. As a young scientist, I think research is where all the ideas come from. And so it is essential that we continue to progress research and to medical science and beyond. In Australia, we need more momentum to have a good value as a country, and so that our healthcare system does not lag behind our Western competitors. Our genetic testing is not as good nor as accessible as it should be, which needs to be improved. More and more evidence is becoming mounted against supporting the argument that genetic testing isn't only pertinent for diagnosis, but it is also very important for prevention, which is always the better thing, prevention over treatment. I will now give an opportunity for any participants to ask any questions they would like. Let's see. Yes, just timekeeping.
Maybe if I could just share that, when we do make some of these massive progresses, like Fabien was able to, with the PhD candidate, make a lot of progress happen. I think my just a thought I would share is that for the broader community, there's a lot of good work happening, but at the same time, all of it needs a lot of support. while compared to maybe 5 years, 10 years back, like potentially, and coming back to the current state, I think current state, the technology is enabling a lot of things, but still there is concentrated effort that's needed to make some of these wins happen. There's investments needed to make these things happen. So Fabien, do our best to keep supporting more researchers.
Yes, yes. So yeah, these are our social medias and things like that. So you can find us there. Yeah, or you can connect with any of us on LinkedIn if you wish. Yeah. So I'll just leave this Zoom open. So if people want to ask questions, I'll probably close it in 5 minutes or so if no one has any more questions or comments. Vivek, do you have anything final you would like to say?
And I just encourage Neeraj and Fabien, like maybe Fabien, like now listening to Neeraj's presentation as well. Like, did you have any thoughts on what Neeraj was sharing, or Neeraj, like on Fabien's specific shares? Did you have any thoughts or comments?
I actually had a question for Neeraj. I was just wondering if your biomarker platform, how does it materialize? Do you intend to make it as an app that any patient can download on their mobile phone, or how does it actually, maybe it's already like that, how does it actually materialize? How do you implement your biomarker platform?
So what is the referral pipeline?
Yeah, so for us, so it is currently configured or customized for that disease area or therapeutic area. So for us now, it materializes as an app, but certain diseases we know could have just the voice biomarkers. So there is a rich repository of voice data coming in through various devices, and not just smartphone, looking at Alexa and other Siri technologies. If we are able to narrow down for certain disease that voice biomarkers have highest sensitivity and specificity, that could look like something like a telephone call, for example, in the future, where you just have a call and you get screened for that disease. So yes, we're starting with the app at the moment, but for certain biomarkers, it's really easy to scale. For example, voice biomarkers. And the referral process, answering to your question, Vivek, is we are controlling currently. It's going through clinicians because we want them to shortlist and select who are the right patients for this, with the hope that once the technology is mature, it can be direct to consumer as well.
Maybe just follow-up question to that, like, when the Dementia Research Center was open, we had the inauguration of the Dementia Research Center at Macquarie. I believe one of the researchers on it was there was some work being done on human biomarkers as predictive disease predictors. So, like, potentially in 5 years' time, we might know that a 40-year-old, when he gets 80, may have dementia. There are some early.
So I'm sure Fabien probably knows a lot in that area, but based on our research, what we have found, there are different streams of biomarker research going on. We are focused on digital biomarkers, which is one stream. And why we chose that is because of its scalability and low cost, whereas there are other streams where people are looking at blood-based biomarkers, cellular-based biomarkers, even looking at brain structure, looking at MRI images to create those biomarker. And there's a lot of technology know-how which goes into those biomarker creations. And I think, like you say, it takes a village to bring together a good solution. And that's what is happening currently. People are trying different areas and are like looking at problem from different angles to find a solution.
I think it's actually really relevant for FOXG1 as well, because there's one thing that I did not mention, that is the initiative from the FOXG1 Research Foundation, which is the American branch of the FOXG1 Foundation. What they did is really important. They built a registry, so a biobank, a digital biobank, and all the families can freely and should freely deposit all the data that they want. That obviously includes the genetic reports, but also they have the ability of sending samples. So skin biopsy or blood biopsy, for instance, and that will be reprogrammed and give iPS cells, so induced pluripotent stem cells, which are models that we, after that in the lab, use as well. But just to go back to simply the registry and the importance of what's called metadata, so the ability to really put together a humongous amount of data. I have been lucky enough to access this registry, and I can tell you that simply looking at the registry, I could foresee some questions that I would not have otherwise thought of, and for instance, very simple things, but in terms of transmissibility, because most of the FOXG1 cases are what we call de novo mutations, where the parents do not carry the mutations. But in very, very rare cases, the parents actually do. So that raised the question of inheriting the genetic mutations, and if so, how? And given that in most of the cases, this is not a disease that is inherited, why? So if you address these questions, then I think this really will help the research, and finding a cure requires this sort of information, and this is really precious information that I got simply by the families coming together and being willing to deposit these data in a biobank or in a registry. So the power of this metadata, which is collecting many different data around the world and trying to get the information out of it, is now really powerful, but it requires the participation of the families and the scientists and everyone, as I was saying before. But this is, once again, a way of accelerating the research towards a cure. So at every level, really, this concept of biomarker and biobank can help, for sure.
Thanks, Fabien, for sharing. I think collaboration is happening so much everywhere, but also, the significance of, even just finding out that it is an autosomal dominant, potentially an autosomal dominant Mendelian disorder, then being put onto the NIPD panels and things. So, and as the costs of diagnostic testing is reducing, I think when Kush was diagnosed, in Australia, FOXG1 was put on the epilepsy encephalopathy panel. So certainly, after the genetic basis of these disorders, the more information that's coming out it gives us a lot more emphasis on broadening the capture range of the syndrome as well, and potentially, like you've shared multiple times, that sometimes the phenotype is not 100% visible or present, but then if it's an autosomal dominant, then it could be stopped before someone gets to that suffering stage. So critical to have some of these super important concepts come together, and I think just technology making it happen is just such a enabler.
Yeah, absolutely, Vivek. I just had a question where, from your view and maybe Fabien's view, what are the biggest, maybe one or two challenges currently in order to, engage and continue further this work?
Yeah, I can definitely find you two because there are quite a few, but the main ones are definitely funding, as it is always with rare, so-called rare genetic disorder. I applied for government funding, the same scheme that would be typically funded for, as I mentioned before, these so-called mainstream disorders like dementia in general. And FOXG1 is imagine that in competition with mainstream funding, which is be it cancer or things like that. Yeah. So the likeliness and the odds of being funded is really low, and that's really a struggle. The entire project has been done with very little money, and as you know, research is very, very expensive. So I think funding is really the main limitation. But let's put that aside and imagine that we have unlimited funding tomorrow. I think the actual limitation in terms of therapy is going to be the delivery method, because, as I mentioned, FOXG1 is essentially expressed in neuronal cells, so brain and spinal cord. And the question is, even if you have a really great candidate, how do you actually deliver that to essentially the entire brain? You can certainly do local delivery, as it is on other disorders like Batten disease, etc., with a small device that is implanted behind the ear. But for widespread pan-neuronal access to the neurons, that is quite a challenge. And the delivery method is also a very active way of research, basic research. And that will also be any breakthrough in this direction will benefit disorders such as FOXG1 for sure. So these are, in my opinion, the main two limitations.
Thanks, Fabien. Also, maybe diagnostics and early diagnostics would help, in early diagnostics and intervention. So sooner that like for instance Canva or like for instance SMA, where the therapy is available, if even if the phenotypes, the seizures are able to be stopped earlier, possibly it's better outcomes. But yeah, conscious of time. Thank you, Neeraj, for some interesting questions. Thanks, Fabien, for such so much of focused work to get to. We didn't touch about Taysha, the gene therapy pharmaceutical company in the US, is already working on FOXG1 as one of the 17 projects that they're working on. So if there's like 9,000 rare diseases, 8,000 rare diseases, if the current trends are that pharma is picking up cures on gene therapy for FOXG1, then certainly it's a good horse to bet on. Like I think it's a and I'm so grateful to like Fabien, yourself, and so many researchers, Alex, yourself, Macquarie, you're a researcher as well. Poonam's got some background around research, but all the volunteers as well, all supporters, everybody who's supported the thing. Alex, I'll let you wrap up. Lillian, thanks for the timekeeping.
Yeah, thank you, Lillian. I mean, I think you wrapped it up perfectly, Vivek. But yeah, I understand that it is a lot about funding, what affects the most amount of people. But something like FOXG1 is so interesting because of all the effects it can have from a single base pair mutation, which I think what sets it aside from other genetic disorders. Yeah, so thank you so much for coming, guys. Yeah, everyone, I hope you have a great week and look after yourself. Thank you.
Thank you very much. See you later.
Thanks, Fabien. Thanks, Poonam.